So welcome to the MSK Science Spotlight. I'm Kevan Kashari, and this seminar series features lectures from today's leaders in basic and translational biomedical science here at MSK. And if you have questions throughout the talk, you can email them to the Office of Scientific Education and Training at the email address oset@mskcc.org or by tweeting at msk-education. You can see that on the slide. Questions will then be answered at the end of the program. For the best viewing experience, please use Google Chrome. If you're using Internet Explorer, please use version 11 or higher. If you experience technical issues during the live stream, please lower the video resolution. That should fix it. And you can do that by clicking on the gear shift on the lower right of the screen. So it's my pleasure to introduce Derek Tan. He's the chair of our chemical biology program at Sloan Kettering Institute, and he's also the director of the transitional PhD program in chemical biology. His talk today is entitled A Drug Discovery Journey from Tuberculosis to Cancer, and we're excited to hear about what's going on in Derek's lab. So take it away.
Thanks very much, Kevan, for the introduction, and thanks for not making fun of me for going to Stanford since you went to Berkeley. I want to thank everybody out there for tuning in, all 7 of you. Oh, 8. I see my mom just logged on. But in all seriousness, I want to start with a couple of personal notes. First, I want to thank all of my colleagues at MSK for doing an absolutely spectacular job throughout the COVID-19 pandemic and the epidemic here in New York City. It has been truly inspiring to be a part of this institution and to see how everyone has responded, especially the frontline clinical staff, of course, but also the essential research staff who have been working hard behind the scenes on COVID testing, to all of the administrative and support staff who make it possible for us to do our jobs. Secondly, as this is one of our first presentations after shutdown academia, shutdown STEM last Wednesday, I wanted to express my strong support for all of our students and their dedication to this critically important mission of eliminating racism, discrimination, and inequality in science. As someone who's worked to promote diversity throughout my entire career, I know how important this is. And I also know that I can do more. So I hope that everyone out there will commit to doing the same so that we can use this momentum to enact real and lasting change. So on to the science. My lab is interested in developing new chemical probes to address a variety of targets that are implicated in cancer as well as infectious disease. And as you'll see today, it's our long-standing interest in tuberculosis that has actually recently led us to a new approach in cancer therapy that we're very excited about. And this work has been done through a collaboration with David Scheinberg as well as Rainier Brenchen's colleagues here at MSK. So to address the challenge of discovering new chemical probes, my lab has long used 2 complementary approaches involving both diversity-oriented synthesis, where we make novel libraries and use them in high-throughput screening to try to identify compounds that can modulate targets, pathways, and phenotypes, as well as rational design, where we use structural and mechanistic information about individual targets of interest to design new chemical probes. And in fact, all of our work in both of these areas has leveraged insights from natural products at multiple levels. So on the diversity-oriented synthesis side, I'll just very briefly show you a few of the classes of compounds that we've worked on over the years, without going into too much detail, but the references are here. We've developed new chemical methods to make 3-dimensional structures in spiroketals, multi-scaffold libraries in alkaloids and terpenoid-like molecules, ring expansion approaches to generate macrocyclic and medium ring structures, as well as most recently a series of palladium catalyze cascade reactions to generate oxygen heterocycles. And I'll note that we have some very exciting hits that have come out of these libraries that we've been pursuing with collaborators, in particular antimalarials and antitrypanosomals that have come out of the alkaloid and terpenoid library. But what I want to focus on today is our work in rational drug design and how that path has led us from tuberculosis back to cancer. So we've long been interested in a mechanistic superfamily of enzymes called adenylate-forming enzymes. These catalyze a biochemical transformation that involves taking a carboxylic acid substrate, attacking the alpha phosphate of ATP, ejecting inorganic pyrophosphate as a leaving group, and generating this acyl adenylate or acyl AMP intermediate. And that intermediate actually has to remain tightly bound to the enzyme because it's highly reactive and readily hydrolyzed. And then typically the same enzyme, often associated with a conformational change, will then catalyze nucleophilic attack on the anhydride to generate an ester, thioester, or amide product. And it turns out that there are at least 9 different classes of proteins comprising 8 different folds that catalyze this same biochemical transformation. So these are found across all 3 domains of life. They impact a huge range of biochemical and biological pathways, and many of them are ripe targets for antibiotic as well as oncology applications. So we have focused largely our efforts on this so-called ANL for acyl-CoA synthetase, non-ribosomal peptide synthetase adenylation domain, and luciferase family. These enzymes all share the same fold, as well as some work on the ubiquitin and ubiquitin-like modifier E1 activating enzymes. But I'll focus on some of our work in the ANL family in particular. So we and others have developed inhibitors of these adenylate-forming enzymes using a very simple inhibitor design strategy that involves mimicking the acyl AMP intermediate with some sort of acyl sulfonyladenosine, as we call it. Now, we weren't the first to pursue this approach. This approach was pioneered by Ashita and co-workers in the early 1990s, who developed the first aminoacyltyrronase synthetase inhibitors, and they in turn were inspired by the structure of this natural product, ascomycin, which they recognize is a close analog of alanine AMP, and in fact an inhibitor of alanine tyrronase synthetase. So we and others, again, have leveraged this approach, and some of the targets that we've gone after are outlined here. I'll mention menaquinone biosynthesis we've targeted in collaboration with Peter Tong at Stony Brook, various natural product biosynthetic pathways that use amino acid adenylation domains we've targeted with Luis Quadri at Brooklyn College, and the ubiquitin family E1 activating enzymes we've targeted in a very fruitful collaboration with Chris Lima here at MSK. But the story I'll focus on is here, salicyl AMS, which is our first generation lead compound to target bacterial siderophore biosynthesis as a novel antibiotic strategy, particularly in tuberculosis. This project was started through a collaboration with Luis Quadri when he was across the street, a colleague at Weill Cornell and now at Brooklyn College. And we've also been working closely with Bill Bashai's lab at Johns Hopkins. So to preface, tuberculosis is actually a global pandemic. We hear a lot about COVID-19 being a global pandemic. TB has long been a pandemic. In fact, there are an estimated 1.7 billion people who are latently infected with the disease worldwide. And the transmission is person to person via respiratory droplets, just like COVID-19. And as few as 10 cells can be enough to infect an individual. So each year, there's about 10 million active cases. So between 10 and 20% of people with latent infections developed active disease during their lifetime. And this leads to 1.5 million deaths in 2018, which is when the last WHO data is from. There is a cure for drug-susceptible TB, and that's a 4-drug cocktail. It's cheap. The drugs themselves only cost $40, and the treatment costs, direct treatment costs outside the US cost about $1,000. Inside the US costs about $19,000. And there's an 85% treatment success rate. The failures come from the fact that this cocktail carries significant side effects and has to be taken over a 6-month period. So non-compliance leads to the development of multi-drug resistant or MDR TB. There are hundreds of thousands of cases of this worldwide. The treatment costs are higher, the success rates in treatment are lower, and the fatality rates are start to get significant. And non-compliance or failure of MDR TB therapy, which can take 18 to 24 months, has then led to the emergence of extensively drug-resistant or XDR TB. This is an emerging infection, very high treatment costs, lower success rates, and high fatality rates. So the important thing to recognize in TB is that most of the resistance we see to individual drugs is mechanism specific. So there are mutations to the drug targets or activating enzymes that convert prodrugs to the active drugs. And so this suggests that drugs with new mechanisms of action should be able to have an impact on MDR and XDR TB. And in fact, bedaquiline, or Certuo, the first new TB drug developed in over 40 years, was approved in 2012, but only for MDR TB. And there was some controversy over this approval. It was based on clearance of TB in sputum samples, but in fact, in the phase 2 clinical trial that was the basis for the approval, there was actually a higher death rate in the bedaquiline arm than in the placebo arm. So clearly, there's still more room for improvement here. So we became interested in trying to target the process of iron uptake in TB through our work with Luis. So iron is an essential nutrient. It's required for all life except lactobacteria, and it's required for proliferation and virulence of pathogenic bacteria. Now, we have very low iron concentrations or free iron concentrations in us, and so to overcome this, bacteria, pathogenic bacteria, biosynthesize natural products called siderophores, which are exceptionally strong iron chelators. And these are released into the host milieu. They rip iron off of primarily transferrins and are re-imported into the bacteria as this octahedral chelate. And then the iron is released by some mechanisms which have been defined, others which have not been defined for use in the bacteria's own redox processes. Now, what was known at the outset of our work was that when you knock out siderophore biosynthesis or transport genes in bacteria, you get strains that are drastically reduced in virulence or even essentially avirulent in animal infection models. And so this suggested that this would be a good target for us to try to go after. So the first step in the biosynthesis of two important siderophores in tuberculosis called mycobactin and Yersinia bactin in Yersinia pestis, the plague bacteria, the first steps are catalyzed by an adenylate-forming enzyme in the category of salicylate adenylation enzyme. And so these use salicylic acid as a substrate. They condense it with ATP to form this tightly bound salicyl AMP intermediate, and then they condense with a thiol and an aryl carrier protein to load this salicyl group onto an assembly line, which then stitches together various amino acid and polyketide building blocks to generate these so-called aryl-capped siderophores. And so again, we focused on this tightly bound salicyl AMP intermediate. And in fact, we had some structural information to leverage. Marr, Heel, and Stubbs had published the structure of this closely related adenylation enzyme, which instead of activating salicylic acid, activates 2,3-dihydroxybenzoate. So it just has an extra hydroxyl group here and a couple of minor differences in the active site. So we knew approximately how these things would bind and what the active site residues were, and importantly, the structure of the intermediate that we wanted to mimic. So we made this very simple change of phosphate in salicyl AMP to sulfamate in salicyl AMS. This is our first generation inhibitor, actually the last compound that I made in the lab with my own hands. And working with Julian Ferreras in Luis's group, Jae-Sang Ryu in my group, who was a postdoc at the time and is now a professor at Awa Women's University in Korea, worked together to demonstrate biochemical inhibition of the salicylate adenylation enzymes. Salicyl AMS has an IC50 in the 10 to 15 nanomolar range. That's pretty good, especially considering there's only 20 nanomolar enzyme in this assay. So 10 nanomolar is as good as you can get for an IC50. And so these are so-called tight binding inhibitors. You have to use the Morrison equation to calculate KIs, and those come out in the low to sub nanomolar range in a mixed mode of inhibition, competitive versus ATP and non-competitive versus salicylic acid. We then showed that you could block production of C14 labeled siderophores in TB using the compound comparable to what is seen in a knockout strain, and further that in cell culture media, we could inhibit growth selectively in iron deficient media that mimic the host environment. In iron replete media, ostensibly growth is not dependent on siderophore production because there are low affinity iron uptake pathways that can take over. We do see some inhibition still that could be due to off-target effects. It could also be consistent with the fact that the knockout is still not completely happy for growth in iron replete media. So micromolar range, IC50s here in liquid culture. Subsequently, Courtney Aldridge's group at the University of Minnesota did a tremendous amount of work to flesh out the SAR around salicylic AMS. And they, in fact, retested the compound under slightly different culture conditions, getting an MIC in the sub-micromolar range. So ultimately, we moved on to collaborate with Bill Bashai and move this compound forward into a mouse model of tuberculosis. In the initial pharmacokinetic or PK studies, we showed that we got good lung exposure, about half the AUC that in the lung compared to the plasma, but short half-lives on the order of 15 minutes and no oral availability of the compound. Moving forward into the efficacy study, what we do is infect the mice on day 0 with about 3 log CFUs of TB. That grows up to about 8 logs on day 14. And you can see when we treat with 5 or 16 mg per kg of salicylic AMS, IP injection daily, 5 days a week, we can get about a 1 log reduction in the CFUs on day 14. So that's a 90% reduction, which would be great in cancer, but in infectious disease, we think on a log scale. And so we really want to see about a 3 log reduction before we start getting excited about this. The next thing we would want to do, of course, is to dose escalate and try to treat for longer periods. Unfortunately, we were precluded from doing that due to toxicity that we observed in the mice. And you'll see that here in the 16 mg per kg group, these mice actually don't survive to the end of the 4 weeks. And these mice at 5.6 mg per kg were starting to get sick. And importantly, they're not getting sick from the TB. So what we see here is that untreated mice can survive the whole 4-week trial in this experiment. But when we have uninfected mice in brown who are treated at 50 mg per kg, higher than the last 2 doses I showed you, those mice actually die within 7 to 10 days. So very toxic side effects here. And the mice also lose weight. And so what we did first was to look at whether there was some cytotoxicity of the compound. But in fact, against a variety of cell lines that we tested, we saw no cytotoxicity of salicylic AMS. Now we did note, Bill's lab noted that in the PK studies, the mice tended to get a black eye on the side where they were injected at 50 mg per kg. And so we wondered, one of the side effects of some antibiotics is that you see hemolysis of red blood cells. So we actually tested that, and there's absolutely no hemolysis that we could detect of red blood cells in this assay. So we had to dig deeper to try to figure out what was going on. And so Chung Ji, who was a postdoc in the lab now at AbbVie, worked with Alyssa DeSankina and our anti-tumor assessment core facility here at Memorial to try and study this further. And so one of the things they did was to do a pilot toxicology study of salicylic AMS to try and characterize the toxicity further. Interestingly, what we saw was that at 6 and 16 mg per kg, we actually saw no toxicity that we could detect either in gross phenotype or in hematology or clinical chemistry at the end of the 2-week period. At 50 mg per kg, we did see some weight loss, but the mice all survived, in contrast to what we saw in the efficacy study. And when we went up to 75 mg per kg on a single dose, we can reproduce that black eye, which the real scientists call a periocular hemorrhage. And in fact, when we did the pathology, that was traced back to facial vein thrombosis. So we're interested by the fact that we saw differing levels of toxicity in this study compared to what we had seen in the efficacy study with a slightly different batch of compound. And so that batch-to-batch variability suggested that it might not be salicylic AMS that was the toxic species, but some fragment or impurity or metabolite. And so we considered what those fragments might be. If you were to cleave salicylic AMS on this red line here, you can get AMS, which is actually a known highly cytotoxic compound and a natural product in its own right, as well as salicylate and salicylamide. In contrast, or alternatively, if this N3 group of the adenine ring is known to potentially attack the 5' position of the ribose to generate a cycloadenosine and a salicyl sulfamate byproduct. And in fact, when we went back to the original efficacy lot and looked for these by LCMS, we can detect them. So I said, aha, it's got to be cycloadenosine because this is a potential alkylating agent, and that's probably toxic. So the next thing we did was went back and looked at our mouse PK samples to see if we could detect these in vivo. And as it turned out, the cycloadenosine was actually cleared very rapidly in under an hour, whereas the salicyl sulfamate actually was retained at low levels, about 0.6 micromolar, throughout the first 8 hours that we looked. So I said, aha, okay, it's salicyl sulfamate. So Chung made these fragments, and we worked with Alyssa to put them into the mouse toxicology study. And lo and behold, salicyl sulfamate has absolutely no effect. It's completely non-toxic, as is cycloadenosine, up to 140 mg per kg, respectively. And importantly, salicyl AMS in this case, we did not start to see toxicity until we escalated all the way up to 150 mg per kg. And this was yet another slightly different batch of the compound, so more batch-to-batch variability. So this really had us scratching our heads. But Chung had the idea that maybe the toxicity we're seeing was due to AMS. And I said, you know, Chung, yes, you're right. It's a highly cytotoxic compound, but we don't detect it. We don't see it in LCMS or NMR, so it can't be that. But Chung said, well, let's test it. And so we put it into the mouse model, and consistent with what had been reported some 30, 40 years ago in the literature, it's a very toxic compound. 0.5 mg per kg daily dosing will kill mice within about 4 days. So what Chung decided to do then was to look for this AMS prospectively. So he realized we might not detect very low levels by NMR or LCMS, the typical methods that we use to characterize purity of our samples. So he went to look for it by LCMS-MS, and in fact found very low levels, less than 1%, in all of the lots that we had been studying, the efficacy lot as well as both toxicology lots. And in fact, when you look at these percentages and multiply them out, the amount of AMS that we are presumably injecting into the mice is consistent with the gradation of toxicity that we see. So this really indicated that AMS was the toxic species. And in fact, we have additional data that indicates it's not a metabolite or a decomposition product. It's actually an impurity that was carried through from an earlier step of the synthesis, and we just didn't see it. So we have figured out ways through the efforts of other people in the lab to get rid of that AMS, to take it down to almost undetectable levels, and we think we can make this compound safe as a result of that. Another approach to doing that, of course, is to make an analog where the salicyl AMS compound is still active, but the AMS fragment is less toxic. And that's been the focus of the thesis work of Lisa Stanke, who's a pharmacology graduate student in the lab. So Lisa put together a really comprehensive SAR study where we could leverage SAR data that had come out of Courtney Aldrich's lab for the salicyl AMS analogs and look at the toxicity of the corresponding AMS fragments. And I won't go into the details here. Suffice to say, many of the changes that we make basically abrogate or drastically reduce the cytotoxicity of the compound, and we can do that without significantly impacting the activity of the salicyl AMS analog in the biochemical assay as well as in the antibacterial assay against TB. When we plot these out, we have the salicyl AMS activity against MBTA, the salicylated adenylation enzyme on the y-axis, and the toxicity of the corresponding AMS fragment on the x-axis. AMS is down here. It's very, salicyl AMS is very potent inhibitor, but AMS is a very cytotoxic compound. And so what we'd like to do is move this over to the right. And so we see some compounds over here that look quite promising in terms of their profile. These are the ones that we have scaled up and are now moving forward into the mouse model with Bill Bashai's lab. And we're looking forward to testing these at higher doses and seeing if we can improve the efficacy further. So we're excited to continue moving that TB project forward. As I mentioned, this had actually given us some inspiration to think about how we might leverage these findings in cancer. AMS is a very highly cytotoxic agent, and in fact, it has been tested in the NCI 60 cancer cell line screen, and it wipes out everything. It has a mean GI50 of 1.8 nanomolar, and it doesn't care where the cancer comes from. It will kill all of them. So the question is, how could we harness that very potent cytotoxicity? So, of course, in the last 10 or 15 years, there's been a tremendous resurgence in interest in antibody drug conjugates. These are ways to use antibodies to target small molecule drugs selectively to tumor sites, where the drugs are then typically cleaved through processing in the lysosome, and then they kill the cell that has taken up the antibody. So this allows you to use highly toxic drugs, but there are some limitations, one of which is chemically we can only fit about 2 to 4 drug molecules per antibody. So you're not delivering many drugs for your antibody. And the production is non-trivial as well. You have to use protein chemistry to couple these things and do that in a robust, reproducible fashion. So we're intrigued by this additional approach that's been explored in the literature called ADEPT. This is antibody-directed enzyme prodrug therapy. The idea here is that instead of delivering the drug, you use an antibody to deliver an enzyme that can convert a non-toxic prodrug, such as the one shown here, into an active drug selectively at the site of a tumor. And this approach is advanced to the clinic in the UK, largely through the work of Caroline Springer and others, where they use this mustard protected prodrug or glutamate protected mustard prodrug that can be cleaved with a bacterial enzyme called CpG2, carboxypeptidase glutamate 2. That cleavage at the N terminus of the C-terminal glutamate releases a carbonic acid intermediate, which then spontaneously decarboxylates to generate the active mustard species. And so, as I said, this was advanced to the clinic in the UK. There, however, were some limitations. So they saw insufficient tumor to blood ratios, which they had to clear with a second antibody. And there was also some immune response to the CpG2, which is a bacterial enzyme. So we can get high amplification of the drug in this ADEPT approach, but insufficient targeting remains an obstacle to the efficacy of this approach. So being here at Memorial, we've been, of course, very interested and excited to follow the work on CAR T cells, chimeric antigen receptor T cells. And much of the pioneering work in that area has been done by my colleagues, Michelle Sadelain and Rainier Brenchens. So the idea here, I'm sure most of you out there are familiar with this, but for those who are not, you take T cells from the patient, infect it with a viral vector that causes the T cell to express an artificial chimeric antigen receptor that's comprised of an scFv targeting domain that recognizes a tumor antigen, a co-stimulatory domain, and a signaling domain. These work together to activate the T cell immune response to the tumor. And there are, of course, 2 approved drugs now based on this CAR T cell therapy approach. Despite these very exciting advances in immunotherapy, there remain some limitations of CAR T cells. In particular, there are antigen-negative cells in heterogeneous tumors that typically escape detection and therapy, and these are what ultimately lead to relapse. There's also this phenomenon of T cell exhaustion, where the T cells are still around, but they no longer proliferate, no longer produce cytokines, and no longer activate the immune response. The tumor microenvironment also is immunosuppressive, so that can repress the immune response. And T cells typically don't penetrate into solid tumors very well, and so the applications thus far have been limited to liquid tumors. And then finally, of course, there have been some cases of severe toxicity in response to these CAR T cell therapies. So we wondered if we could perhaps combine some of the activities of a small molecule drug with the CAR T cells in an innovative strategy that we've developed with David Scheinberg's lab. So this class of CAR T cells is envisioned to be basically a micropharmacy that could deliver or generate or produce or unmask small molecule drugs locally at the site of a tumor, in addition to the immunotherapeutic effects. So David's great at acronyms, so he calls these seeker cells, synthetic enzyme-armed killers. And the idea is that these would, like any other CAR T cell, would home in on a tumor, recognize a tumor antigen, and proliferate locally. We would then administer an inactive, non-toxic prodrug systemically, as one would do in ADEPT therapy. But now the seeker cell has proliferated, and each of those cells is also producing multiple copies of the activating enzyme. So we get 2 more levels of amplification in addition to the catalytic activation of that prodrug to form the active drug, hopefully in very high local concentrations at the tumor site. So there are a lot of attractive features of this approach. As with ADEPT and ADCs, we can use highly toxic drugs and we get some amplification of those drugs. But in addition, as I noted, we would get amplification via the cell proliferation as well as the expression of the enzyme by each cell. Furthermore, these small molecule drugs can kill antigen-negative variants and bystander killing. They can diffuse and penetrate into solid tumors. This is a modular platform that we can envision using with various prodrugs and enzymes. That enzyme expression, if necessary, we could make it gated upon antigen recognition to improve selectivity, although that hasn't proven necessary. And we can envision dose sparing of the CAR T cells to possibly reduce some of the risks of toxicity. So lots of attractive features, but also many potential challenges. We have to figure out how to express these bacterial enzymes in human T cells. Some of them are fairly large, larger than normally seen in CAR constructs. We're worried that maybe the drug would be toxic to the CAR T cell that produces it, or that the enzyme itself would be immunogenic, as has been seen in ADEPT, and that might limit the efficacy of the approach or the activity of the enzyme in particular. We also had to worry about the pharmacokinetics, coordinating administration of both the cells and the prodrugs at the right times. And finally, if the drugs were activated locally but diffused away too far, then we could suffer from off-tumor toxicity. The work on the chemistry side of this has been done in my lab, primarily by Peter Lee, who's a graduate student in our tri-institutional PhD program in chemical biology, as well as Nihar Kanaravala, who was a postdoc in the lab. And the idea here, again, is that we would have a potent drug that we could mask with some kind of protecting group, if necessary, linked via some self-immolating linker to allow the chemistry to happen. And we could cleave the mask off with an enzyme, an unmasking enzyme or activating enzyme to generate the active drug. And so this is a modular platform. We envision being able to do this with a variety of different classes of drugs as well as different masking groups. The drug classes that we've looked at so far are AMS, of course, the cytotoxic natural product, the nitrogen mustard ZD2767, which was used in ADEPT clinical trials, and this targeted kinase inhibitor, which is an analog of gefitinib called APDMG that was developed by Barney Yu in Michelle Bradbury's lab here at MSK. And we used two masking strategies. One, the glutamate masking strategy that was used in ADEPT using bacterial CpG2 to cleave. And second, a cephalosporin masking group that can be cleaved using beta-lactamase. So the complete prodrug structures are shown here. This is one of the compounds I'll tell you about, AMS glue, and another one, Ceph-AMS. In AMS glue, the bacterial enzyme CpG2 cleaves, again, N-terminally to the glutamate, and then that carbamic acid intermediate spontaneously decomposes to give us our active AMS compound locally at the tumor site, hopefully. In the case of beta-lactamase, this will open the beta-lactam ring, and that sets up for a spontaneous elimination across this allylamine to eject the sulfamate leaving group, thereby generating AMS. So two different ways to unmask AMS from these two different prodrugs, depending on which enzyme we use. And we also developed the mustard prodrug and the gefitinib prodrug based on the glutamate masking strategy for use with CpG2. I won't show you that data, but we've advanced those compounds through this process as well. So this work to move these compounds forward into the cellular and ultimately animal models has largely been done by Tom Gardner, a fantastically talented postdoc in David's lab, along with Chris Bourne, another graduate student who's been working on the project and made major contributions. So in this first slide, what I show you is demonstrating that the AMS glued prodrug can be activated to form AMS, so it's accepted as a substrate by the enzyme, and that activation induces cancer cell apoptosis. So in the first panel on the bottom left, you can see in this glutamate release assay using Amplex Red that CpG2 does in fact cleave the glutamate off of AMS. So it's accepting this prodrug as a substrate. Tom showed that AMS glue is basically non-toxic to this SET2 leukemia cell line, 30,000 nanomolar IC50 at least, if not more, probably more, whereas AMS is very highly cytotoxic with a 15 nanomolar IC50. So a selectivity index of at least 2,000-fold there. And then Tom showed that in cell culture, if you treat SET2 cells, again, with AMS glue, you get no cytotoxicity. If you treat with AMS, highly cytotoxic, this is at 1 micromolar concentration, so way above the IC50. And if we treat with the prodrug plus CpG2 recombinant enzyme, we can reproduce that cytotoxicity of AMS. So we've activated the prodrug and to form AMS and activate its cytotoxic activity. The next work that Tom did was some model studies in HEK293Ts. I won't show you that work. I'll just jump forward to his work in seeker cells, which are derived from primary human T cells. And he infected them with this construct, which he calls the CpG2-19BBZ seeker construct. So it's got the CpG2 cleaving enzyme. It's also got an anti-CD19 targeting element, a 4,1-BB co-stimulatory domain, and a CD3 zeta chain signaling domain. So it's got the complete construct that one would need in a potential therapeutic cell. And so the idea here was, again, to show that the enzyme CpG2 could be expressed in the seeker cell, could activate the prodrug to the active drug, and that would induce cancer cell apoptosis. So in the first panel, we see SET2 cells, which are antigen negative cells, actually. They don't have CD19. We can, if we treat with the seeker alone, there's no activity. And if we add the prodrug, now we get very potent cell killing. In this second panel, we show synergy between the seeker cell and the prodrug. So these are Raji cells, which are antigen positive. So high enough concentrations of the seeker cell can result in cell killing of the Rajis. But you can see even at these very low concentrations of the seeker cells, where the cell alone is not very effective, adding the AMS glue prodrug really activates, increases the activity significantly. And finally, in this panel, in this FACS analysis, it's a very intriguing and important study that Tom did. We looked at a mixed tumor or a mixed cancer cell culture of Raji cells, which are CD19 positive, and SET2 cells, which are CD19 negative. You can see that the seeker cells only kill the Rajis. They don't fully kill the SET2s. There's a little bleed over there. The prodrug alone does nothing, but when we add both of those together, we can kill both sets of CD19 positive as well as CD19 negative cells. Interestingly here, we also see that the T cells, the seekers, survive. Now this is a short time point, so as we go to longer time points, the seekers do start to get sick, but there are some interesting ideas here about why the seeker cells may be a little bit resistant, and we're examining that further. But regardless, we're happy to see this differential cell killing because it bodes well for what we want to do in vivo. These are the results for AMS glue. We saw similar results with Ceph-AMS and the beta-lac cleaving enzyme that I won't show you. So now moving on to the tumor model, or a mouse tumor model. The scheme here is that Tom engrafts the tumor on day 0, and then we're going to show you the data for Ceph-AMS. So here the seekers, these are now the beta-lactamase seekers, get administered on day 2. And when those have expanded to high levels at the tumor, which Tom tracked using a chemiluminescent variant of the seeker, we go ahead and administer the prodrug and then see what happens after 8 days through bioluminescent imaging of tumors that are engineered to express firefly luciferase or Gaussian luciferase. So in this first panel, there's 6 groups here, and so I'll take you through it step by step, but basically this is showing the synergy between the seeker cells and the Ceph-AMS prodrug. So the top row are untreated, no cells, no prodrugs, and you can see imaging of the tumors. And when you add the cells alone, this is a subtherapeutic dose of the cells. Of course, you can use a higher dose and kill these tumors completely. We wanted to see the synergy. So this is a subtherapeutic dose. You see some reduction in tumor imaging in both cases. But when we add Ceph-AMS, the prodrug, now we can completely clear those tumors, only on the seeker cell side, not with conventional CAR T cells, 19BBZ CAR T cells. So this was a very encouraging result for us. And we also saw similar effects with AMS glue and the CpG2 cleaving enzyme. Now, importantly, Tom also showed that the seeker activating enzymes are still active even after the seekers are exhausted. The T cell immune functions are no longer there. So this is after 26 days. You can see these seekers are producing markers for T cell exhaustion. They no longer exert the immunologic effects. But here on day 21, we can re-administer more prodrug and get a small decrease in the amount of tumor, indicating that there is still active enzyme around and potentially being produced, presumably being produced by the cells, even though they no longer have the T cell functions. So Tom then went on to look at activity against antigen-negative cells in a heterogeneous tumor model. So what he did here was take NALM6 cells, half of which in which the CD19 antigen was knocked out by CRISPR, and he put Gaussian luciferase in the CD19 positive cells, Firefly luciferase in the CD19 negative cells, so we could track them separately. And so the negative cells are shown here, but untreated cells, you see some of the antigen negative tumor around. The seeker cells alone don't do much of this, but when you add the Ceph-AMS prodrug, we can now start to eliminate those CD19 negative tumors, which are in the same place as the CD19 positive tumors, of course. And so that's how the cells are able to traffic there. On the right, you can see some of the work that Chris Bourne did in an immunocompetent syngeneic mouse model, where we wanted to see if we would still have enzyme activity even in an immunocompetent model. The previous models were in NSG mice. And so what Chris did was to engraft with a mouse tumor on day 0, administer mouse-derived seekers on day 7, and then look on day 15 at nitrocephin activity of the beta-lactamase. So this is just a surrogate marker for beta-lactamase activity. And what we can see is even after 8 days, there's significant activity there. We have more recent data that indicates that there is an immune response to the beta-lactamase cleaving enzyme, even though it's been engineered to remove some of the antigenic epitopes. However, that does not seem to compromise the activity of the enzyme. And in fact, we see a bigger antibody response to the CAR construct itself, which is known in the literature, than to the beta-lactamase. So it seems like this could have some legs to it. Finally, Tom looked at a solid tumor model, which is a subcutaneous model, just using these Raji CD19 positive cells, and showed that we do get a survival benefit with the seeker cells and Ceph-AMS. But most importantly, when we go in and do histology of the tumor to look for beta-lactamase activity, we see these punctate areas, which we think could be the seeker cells, and a halo of beta-lactamase activity around them. And in fact, that activity diffuses throughout the entire tumor. You can see it's brown all the way through, even though it's concentrated around these spots. And in fact, we also were able to excise some of these tumors and looked by LC-MS/MS for AMS, and we detected, in one case, 284 nanomolar, about 10 times higher than the IC50. So we're very excited by these results and looking forward to continuing to move this project forward. So I will close up just by summarizing. We started this journey by interest in tuberculosis. One of the phenomenal things about Sloan Kettering is we're able to go where the science leads us, and our science happened to lead us to infectious disease at that time, but it has in fact come full circle to this new exciting to us approach to cancer therapy involving the seeker cells and targeted micropharmacies. So we're very excited to move both of these projects forward and see what kind of impacts we can have. So I'll close by thanking the people who did the work. You've seen their names and faces as I've gone through the talk. This is my research group. I must say I'm very proud of all of them. We have been existing, like all of you have, online for about a 3-month period, and they really did a tremendous job rolling with the punches and keeping things moving and, in fact, augmenting all their academic studies along the way. So I appreciate all of their work. And finally, I'll put a plug in for our tri-institutional PhD program in chemical biology, which Kayvon mentioned. This is a program of Sloan Kettering, Weill Cornell, and Rockefeller University. There's about 50 faculty across all 3 campuses that students can work with. They have a tremendous track record scientifically, and I can say, based on the last few weeks, that they are also extremely dedicated to promoting diversity and inclusion in science. And I look forward to working hard with them on continuing those efforts going forward. So finally, thanks everybody for your attention. Thanks for coming out, and I'll be happy to take any questions.
Thanks, Derek. That was awesome. So, you know, as the questions are coming in, I'm going to try my best to summarize and ask you like direct questions that take into account a couple. So one of the few that have come in have asked kind of mechanistic questions about how you think AMS is working in cancer cells. So I saw that, I mean, it's very toxic across like a whole range, but what do you think the mechanism is in cancer cells?
Yeah, that's a great question. So there is one report in the literature that, there are a couple of reports in the literature that have tried to probe this. One early report from the 70s suggested it could affect a step in translation, not directly associated with aminoacyltyrranase synthetases, aminoacyltyrranase. But another more recent report showed that AMS can act as a prodrug that gets activated by E1 activating enzymes, actually. And so Millennium has a compound, now Takeda, MLN4924, which is a NET8 E1 inhibitor that advances to the clinic. And it turns out that's an analog of AMS, and it turns out that acts by undergoing a reverse reaction with a thioester product of the E1 to form an acyl AMS type intermediate that's the actual inhibitor of the E1. So the Millennium group suggested that could be a mechanism of toxicity of AMS. That is certainly one possibility. We've been interested in trying to think about other pathways that might be impacted and that might be more globally important. And so those studies are ongoing, but it's an important question for us and a tough one, because any modification we would make to the prodrug to try and put an affinity handle on actually kills not to the prodrug, to AMS, actually kills the cytotoxicity. So we have to do it by sort of functional analysis.
So just kind of building on that, why do you think or maybe it's related to the mechanism, which kind of dragged me to that kind of topic, is why do you think it potentially kills the cancer cells faster earlier than it does the seeker cells themselves? Like, meaning like, do you think there's like some relationship between the mechanism and, you know, T cell biology that protects?
Yeah, great question. So we don't actually think that is the particular case, although there are some examples where we could envision using drugs that are not active against T cells if that becomes an issue. But we know that in a regular cell culture cytotoxicity assay, this thing will kill T cells just as well as everything else. I think it's a timing effect there, maybe a proliferation effect of the activated seekers, but we're still investigating that.
So you mentioned that there's some immunogenicity of the beta-lactamase enzyme. So are you thinking that maybe you could explore mammalian analogs that maybe aren't as immunogenic as the current state lactamase that you're using?
Yeah, that's a great question. So I think the first thing to recognize is there are actually myriad therapeutics that are bacterial enzymes. And so this immunogenicity question is not a new one, but it is not necessarily one that inactivates those drugs. Some of them are quite successful. However, it is absolutely something that we've thought about. The first thing we've done is to try to make versions of CpG2 and beta-lactamase that have been engineered already in the literature to remove immunogenic epitopes. And so those are the ones that we've been using. But beyond that, we have been thinking about and investigating the use of human enzymes that might be able to carry out some chemistry that we're interested in. Now, the challenge there, of course, is you have to worry about off-tumor, off-seeker activation of the drug. But we have something that looks interesting, and we will see how it goes from there. We're also looking at a variety of other bacterial enzymes that do things that are, to me as a chemist, more interesting than cleaving off a protecting group. And so those are things that others in the lab, including Brody Corliss, are working on, and we're very excited to move forward.
Cool. So do you think, this is kind of taking the CAR T kind of in a different direction, do you think it's possible that you could make seeker cells from non-T cells? Meaning, does it have to be a T cell? Could it be a fibroblast that went to the tumor, or macrophage, or endothelial cell? Like, could it be any other cell type that would be the kind of the carrier of the enzyme as opposed to a T cell?
Sure, absolutely. So there are other cell types that one can envision targeting to tumors, and in fact, CDEPT therapy uses C. diff to try to get at certain tumors, and with the same sort of the same sort of enzyme pro-drug strategy. And there is other work that we know about, of course, going on uptown to use bacteria as delivery agents as well. So I think there's many different cells that could potentially be used as delivery agents. As well, there's other applications potentially of CAR T cells and seeker cells beyond cancer, because we could use these cells to deliver other classes of drugs besides cytotoxics. Anything where you'd want a high local concentration on your disease site of interest, wherein systemic administration of the drug carries unacceptable toxicity. Those are the potential applications that we envision in the future.
Maybe just one last question kind of on the action of the pharmacy. So if so you deliver the seeker cells, these T cells to the local area, and then they amplify as a function of the activity of the T cells, and they expand. Again, the obvious combination would be to combine it with immunotherapy and hopefully, block checkpoints. So, do you see that as like kind of the ideal platform, is that you have this proliferating T cell and then you do checkpoint blockade and you just dramatically amplify? And if so, do you think you could increase, do you need to increase the AMS concentration or any of your other kind of toxic agents, or is it enough the way it is?
Yeah, that's a really interesting idea. It's not one that we've pursued thus far, but as far as the last part of your question, do we need to increase the AMS concentration further? So far in the mouse models, we're seeing effects, but we can always do better. The two systems that I showed you, each of them has their pros and cons. So Ceph-AMS is a great substrate for beta-lactamase, but it has a little bit of instability on its own. So we treat at 4 mg per kg to avoid any off-tumor toxicity. AMS glue is completely stable and non-toxic. We treat at 50 mg per kg there, no problem. But it is a lousy substrate for CpG2. So we actually have work going on in the lab to address both of those problems. Peter's developed an AMS glue analog that we think that we've shown actually is a much better substrate for CpG2, and we have an idea for a cephalosporin analog that may be more stable on its own to spontaneous. We think it's spontaneous hydrolysis of the beta-lactam.
Sure. That's awesome. Well, thanks, Derek. I think we're right at the end. So just want to thank you again for that awesome talk. And thank you all for joining us today for Professor Derek Tan's talk. Our next seminar happening this Wednesday, June 17th, will be from Dr. Stephen Long, and we're excited to see him presenting a lecture on and now for something completely different, structure and mechanism in the calcium activated chloride channel, BEST1. And so we hope to see you guys there. And thanks again for joining us.